The phoenix is an iconic bird that symbolizes renewal and resurrection. The phoenix effect is a resurrection of denatured proteins. These specific proteins are then refolded back into their original three-dimensional shape, making them functional again, and in this case, improving their original activity. Proteins are large, complex molecules that play many important roles in the body. They have specific roles that are required for the structure, function, and regulation of the body's tissues and organs. The structure is the major determinant of all the overall functions of the protein. Proteins are chains of amino acids. The amino acid sequence in a polypeptide chain is known as the primary structure of a protein. The primary chain folds up to form a complex three-dimensional shape such as an alpha helix or beta sheet. In a secondary structure, the chains of the strand fold into a coiled structure and will bend and twist around themselves to form a specific three-dimensional shape, which is known as the tertiary structure. The secondary and tertiary structures of a folded protein are directly related to the function of that protein. The most common example of this is hemoglobin, which is a tetramer. It has 4 subunits composed for 2 alpha and 2 beta chains, each containing a heme group. There are 4 binding sites for oxygen on the hemoglobin molecules because each chain contains a heme group. Natural proteins that are exposed to high temperatures, pH, and chemicals causes the alteration in the shape of the protein, making it unnatural or denatured. Proteins must achieve and retain a three-dimensional conformation in order to function properly. If protein folding is disrupted, proteins will become unstable, interact with one another, and aggregate continuously, forming fibers which can be toxic. While misfolded protein diseases contain multiple interactions between the aggregates of proteins, all denatured proteins share a common structural feature known as the amyloid fold. Amyloid folding is the aggregation of misfolded proteins found in between the neurons in the brain. This causes lack of interaction between the neurons. Neurons hardly divide, which allows them to accumulate greater amounts of denatured proteins than other cell types. This accumulation of these denatured proteins leads to various neurological diseases, including mad cow, Alzheimer's, and Parkinson's disease. Prion disease, or mad cow disease, is a gradual neurodegenerative condition. The cause of prion disease is from a buildup of denatured proteins found in the brain. This prion protein undergoes a change in the shape, recruiting other naturally occurring proteins to become denatured, starting a chain reaction. This is the reason why prions are compared to zombies. They are constantly denaturing other proteins, rapidly spreading the disease. Like mad cow disease, researchers are examining aggregations of proteins in Alzheimer's disease, which would explain why this disease worsens over time. Alzheimer's disease is a brain disorder which affects the memory. People with Alzheimer's disease have an increased amount of denatured proteins which form amyloid plaques that affect the memory. Amyloid plaques are found in the spaces between the brain's nerve cells, which inhibits neuron interactions. The plaques are toxic proteins caused by aggregations of beta amyloid structures. The phoenix effect is very important because of the large amount of neurological diseases caused by protein denaturation. Scientists have been working on a way to resurrect denatured proteins to resume their original function. A study was performed by David Avner and Vladimir Vinohradov to demonstrate how this process can be done. In order to allow proteins to become renatured, Avner and Vinohradov used a form of sol-gel transition. The sol-gel transition is a technique used for protein renaturation. Specific proteins were denatured by heat using carbonic anhydrase. The denatured proteins were mixed with a solution with evenly dispersed inorganic nanoparticles. The solution was allowed to sit until it solidified into a gel. The nanoparticles bind together, creating a separation between the individual unfolded proteins, inhibiting them from aggregating and allowing them to refold into their original structure. This study also displayed an increased amount of activity compared to the native protein structure by 180%. This process, however, will only work with unfolded denatured proteins. This project is continuously being developed in hopes that in the future it can be applied in medicine.